All right. Good afternoon, everybody. Thanks for joining us for today's seminar. Um, it is, it's my pleasure to introduce the speaker today, Dawson White. Before I do that, I just want to mention a few logistics. Because this is a hybrid seminar, those of you who are tuning in online, please submit your questions in the Q&A box in Zoom. And if you're in person, please use the mic in the middle aisle to ask your questions, so that those of us who are remote can hear your questions. And if you are on site and you want to join Dawson for lunch in the bistro afterward, you're welcome to do that. Just look for him and Matt Nelson. Okay, I just want to begin by acknowledging that the Field Museum resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, the Odawa, and Potawatomi, that we recognize and are grateful for these original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside in Chicago today. Dawson, yeah, so gosh, there's a lot to say about Dawson. So Dawson was a PhD student at University of Illinois at Chicago, and I served on his dissertation committee. And he came to us. So Dawson is like a real, what I would say is he's a true botanist in basically every sense of the word. And when he came to us, he was really seemed intent on sort of following the footsteps of Tim Plowman, whom you'll hear about today, to carry on his legacy in studying the systematics of coca and its relatives. And early on, Dawson expressed a lot of interest in doing very traditional monographic work, a lot of field work, a lot of careful revisionary morphological systematics. And over the course of his PhD, he really expanded his horizons and embraced the rise of next-gen sequencing. So he did all of the traditional and sort of very rigorous field work and specimen collection and study that traditional systematics requires, but he also embraced next-gen sequencing and did both target capture and rad sequencing for his dissertation. It bears mentioning that doing your PhD on a Schedule II drug plant has its own sort of overhead in terms of dealing with like a lot of red tape, needless to say. And Dawson exhibited really admirable like logistical skills, I would say, in getting in handling all that in his research program. And so over the course of his dissertation and subsequently, he's made some really amazing new discoveries about coca and its wild relatives, which we'll hear about today. His papers on this topic, I think, are really exemplary of integrative collections-based research. It's just a fraction of what's in the pipeline. So, there's a lot to be coming out, soon about the family as a whole, about the coca genome and so on. And I also want to mention that besides, his work on coca, Dawson has really displayed his excellence in research in spectral reflectance. And, he was really the one who kind of spearheaded a lot of interest at the museum, I think, in spectral reflectance. He organized, discussion groups. He got vendors to come in and exhibit equipment and so on. And he's really been a key person in implementing hyperspectral research at the museum, including a project that I hired him for as a postdoc to study reflectance in dry ice in Alaska, which we might hear about today. Yeah, so I just want to, I guess, finish by saying that, oh, and I should also mention, of course, that Dawson's work, extends beyond, just systematics, quote unquote. His work, obviously, with the Centinela flora and, has important implications for conservation. And so, yeah, just I think underscoring his true sort of broad botanical skill set, that he brings to the table. And I just want to finally mention that, as we are very sorry to see him go, but in the fall he will be going to Harvard, moving him and his family to Cambridge, Massachusetts, for a prestigious research fellowship at the Harvard University Herbaria. He will continue his coca research with a focus on spectral reflectance. And yeah, so hopefully he'll be able to work on some of the specimens that Dick Schultes himself collected. And okay, so with that, I will turn it over to Dawson.
All right, thank you, Rick. Yeah, so today I'll be talking about coca, which holds a special place in the Field Museum Herbarium collections, because here we have the world's largest and most important research collection of these coca crops, as well as their wild relatives in the genus Erythroxylum. And certainly a theme of my research has been the utilization of these collections, and with my PhD, and then these postdocs. I've been here for 10 years, and over this time, the developments in technologies and methodologies have really allowed us to unlock a lot more data than we ever have been able to, and really increase the value of these collections, as I'm going to show you here today. But the other theme is trying to answer this question: what is coca? When I tell people that I work on coca, they overwhelmingly think I'm referring to chocolate or cacao, but no, coca is the sacred Andean and Amazonian crop, grown for the stimulant and medicinal properties of its leaves, and of course, it's also the natural source of cocaine. But I certainly believe people should know coca by name, because in the words of Richard Schultes from Harvard, who Rick mentioned, coca is the most important and culturally significant narcotic of South America, and of the New World flora has played the greatest role in Western medicine. So yeah, I think this justifies a bit of an introduction into what this plant is and its really complex and fascinating history. But before getting into that, I'd like to acknowledge some of my collaborators in this human dimension of coca. And this is specifically Carol Konzelman, anthropologist who's done a lot of work in Bolivia. And then Susie Mejia, Daniel Montoya, and their team, who comprise Proyecto Coca, based in Medellin, Colombia. And yeah, Susie and Daniel are artists by trade, and yet they're interested in the same questions I am. But as artists, their methods of investigating, but also communicating results is really innovative and inspiring. So these have been productive collaborations, and also these people take really great pictures, which I'm using throughout this talk. So Carol and I have this review that's still in press, wherein we estimate that over 5 million people in South America are using coca leaves today. And they're using it for medicine, wherein leaves are ingested or poultices are made for topical applications, or there are infusions of the leaves into water or into alcohol for addressing a number of ailments. However, coca is mostly consumed as a stimulant. For travelers to the Andes, you might encounter coca tea. because it is thought to be an effective remedy for altitude sickness. And but there needs to be some more research into that. And then there's chewing the leaves. So coca chewing is a passive process. It's fairly simple. Leaves are, a small handful of leaves will be delicately arranged and placed in the mouth, lightly chewed until macerated, and then just held in the cheek. You can see people doing this in the bottom left image. And then it's held in the cheek for 1 to 2 hours. The juices are just sort of swallowed and absorbed. And then the quid of coca leaves is carefully discarded. And so this gives coca chewers, as you hold the leaves in your mouth, a distinctive bulge in the cheek, which you can see here in this picture of me from 2007. And indeed, I enjoy chewing coca leaves because it is a focused, clear stimulant high, and you can't really overdo it nearly as easily like you can with coffee. And in addition to that, so it also is used for studying by a number of people. But in addition to that, like for instance with field work, which was going on here, where we were climbing this 1,000-meter mountain into cloud forests to make collections and also camera trap Andean spectacled bears, chewing coca leaves made this hike a whole lot easier every day. And so it was an enjoyable ritual activity. But coca also has some interesting metabolic, other physiological and metabolic effects. Specifically, it suppresses glucose metabolism and instead shifts to fatty acid and triglyceride metabolism. So when you're working all day long, this is certainly a preferable method of energy production within the body. And this is due to a complex suite of chemicals whose bioactivity, whose pharmacology is not really well characterized. And in addition, there are a number of nutrients with significant concentrations in coca leaves. Most notably, people have identified calcium as something that has very high concentrations, relatively speaking, and could be an important component of the Andean diet. Lastly, chewing these leaves really doesn't seem to have any negative effect in your mouth or in your body as contrasted with tobacco or khat. And in fact, the opposite may be true. These leaves could possess some anti-carcinogenic effects. So for over 500 years now, people have been writing about how coca leaf takes away hunger and thirst. And yet coca is also used in social gatherings like parties, community meetings, or festivals. And Carol Conselman and other anthropologists have taught us, Westerners, that when chewed collectively, coca inspires communication and facilitates social cohesion. And more deeply, it can really, at least among Andean communities, the Quechua, the Aymara in Bolivia, signifies friendship and can really symbolize community itself. During the time of Spanish contact, from historical accounts, we know that coca was being consumed from Nicaragua down into northern Argentina. Northern Argentina, it's still used there today, and also throughout Amazonia. So while there's not a lot of evidence from Amazonia itself, we know that that coca use was very widespread in Central and South America. And in addition, coca use is very old. The veneration of coca leaves is evidenced by archaeological evidence that has been gathered from up and down the Andes, from Panama into northern Chile. And we have found coca leaves themselves in ritual bundles, as well as coca leaves found in the mouths of mummies. And in addition to that, there are bountiful sculptures, ceramics, artworks, and other paraphernalia. And these evidence an ancient use of coca in ceremonial and spiritual affairs, which is why I say that coca is sacred here. The word coca is actually derived from the Aymara or Quechua word coca, which is the archetypal tree. And coca is said in the Andes to be a gift from Pachamama or Mother Earth because of its beneficial properties. Okay. And some of the most beautiful ceremonial and ornamental artifacts found in South America are associated with coca. Here in the top right, you can see this gold poporo from Colombia, which is a vessel used to hold an additive powder that is mixed with coca leaves in the mouth. It's mixed with this stick that's seen to the left of the gold poporo. And then in that statue, adjacent to that, you can see this person holding the vessel and then also holding this stick and using it for the consumption of coca leaves. So, these ornamental and also just real paraphernalia are very widespread. But the oldest archaeological evidence comes from northern Peru, where actual leaves believed to be the Trujillo variety. And I would really love to see these leaves. I look forward to that moment when it comes. These leaves were found and they're almost 8,000 years old. And in addition to the leaves, down in the bottom here, you can see this gray rock-like thing. This is ypta. This is also an additive for coca. In the bottom right, you can see some scratches, which indicates the flaking off of this ypta. And the powder from it is added to the quid of leaves in the mouth. What's found at this archaeological site is almost exactly what we see in markets today. This image in the top right shows a variety of different yptas, and they are derived from the ashes of various botanical materials. There's an image of quinoa being burnt down here. So this evidence makes coca also one of the oldest crops in the Americas, equivalent with squash, quinoa, and some other root crops. But coca leaves were not always an exotic foreign commodity. I was surprised to learn by reading this book by Paul Gutenberg, who's at Stony Brook University, just how popular coca leaves were in the United States and also in the West. And I really like this quote because in addition to giving us a comparison of the popularity with Coca-Cola, it also brings into view the fact that most of us at this point possibly don't identify that Coca-Cola, the most popular beverage in the world, of course, has its basis, its name in the coca plant itself. And the coca leaves in the West really became popular because of cocaine as a medicine. So it was isolated in 1855 by German chemists because these people had identified the psychoactive compounds of leaves, and then they isolated pure cocaine hydrochloride. It was initially identified as a stimulant. Sigmund Freud has been marked as possibly one of the first cocaine addicts. Here's a quote from Freud about using cocaine. He says, "Exhilaration and lasting euphoria. No craving for further use of cocaine appears after the first or even after repeated taking of the drug." It wasn't until really the turn of the 20th century that the detrimental effects of cocaine use became widely acknowledged. Early on, cocaine was also used as a treatment for morphine addiction. However, really why Schultes had his quote, and the real revolution in medicine coming from cocaine is because of its anesthetic properties. So as a topical anesthetic, it was initially used in surgery and when nerve block sort of protocols were invented, this totally changed the game for surgeries happening. And it's still used today in modern nasal and eye surgeries. It's also a vasoconstrictor. And so some of the synthetics really don't do as good of a job, apparently. And from this popularity of cocaine, there also was a very large coca and cocaine commercial market. And so these products range from cocaine for your hair to toothache drops for children, and hundreds of products. And one of the most popular was a wine, which was an infusion of coca, a coca wine, if you will. And the marketing of this coca wine was something that involved a lot of celebrities, including Ulysses S. Grant, and here, as I show, the Pope himself, Pope Leo VIII. And yeah, historical accounts show that this was just incredibly popular. And another wine that was on the market was made by someone in Atlanta called John Pemberton, but when Prohibition happened, he decided to change up his recipe and he created this. So Coca-Cola is a mixture of natural flavors of the coca leaves and caffeine and from the cola nut of the Cola acuminata tree from Africa. And this beverage was created in 1886. In 1903, the cocaine was dropped from the recipe. And as revealed by Gutenberg, Coca-Cola actually became a heavy political lobby for coca and cocaine regulation. And this is possibly to achieve exemptions for its own trade in the coca leaf, because to this day, Coca-Cola remains one of the very few companies that can internationally trade coca leaves. It gets leaves from northern Peru. As I said, that Trujillo variety. Leaves are brought to the United States, where cocaine is isolated in New Jersey and then sold to the pharmaceutical market. And the natural flavors, specifically wintergreen oil, still goes into Coca-Cola. However, in 2016, there was this headline in the Huffington Post, which would catch your eye, indicating that not all of the cocaine goes into the pharmaceutical company, goes to big pharma, at least initially. So, yeah, it's interesting that that Coca-Cola, one of the most popular beverages in the world, is also to this day almost monopolizing the international trade of coca leaves, and yet the product itself has clearly purposefully distanced itself probably from the coca leaves themselves, certainly from cocaine, right? But ending with that, let's shift more to the scientific aspect of things. And we'll transition by talking with cocaine as in its role for the plants. Cocaine is a natural proven insecticide. It is an octopamine, which is a neurotransmitter in insects. It's an octopamine reuptake inhibitor. The analog of that is dopamine and serotonin in mammals. And so when leaves were sprayed with natural concentrations of cocaine, such as is found in Erythroxylum plants, caterpillars stop eating, they convulse, fireflies will blink faster, and honeybees will dance harder, signifying much a bigger reward than they actually have found. And as further evidence of its role, Schmidt and colleagues, John Dauria and colleagues have characterized the cocaine biosynthesis pathway. They did the final steps in 2015, and they showed that higher concentrations of cocaine are produced in young vegetative tissues and reproductive tissues. So in this image on the bottom right, you can see a young unfurling leaf, and highlighting in yellow the actual fluorescence of the enzymes responsible for making the cocaine, and then below that, an anther sac. So we can see where cocaine concentrations are higher in these plants, and they are in places where things are more vulnerable to herbivory. So the plant itself, Rick talked about me as a botanist extensively, but coca wasn't actually what I planned on studying intensively at the start of my PhD because of the regulations around it and perceived difficulties there. But the initial taxonomy for coca was described in 1786 by Lamarck. He described one single species, Erythroxylum coca. And up until 1860, that's where the taxonomy stood. But as I mentioned, cocaine was isolated in 1855. And after 1860, with leaves coming into the Western market and physicians and pharmacists encountering leaves, wondering what their identity was, the taxonomy jumped up to 14 taxa, most of these things conflicting by 1910. After this time, leaves became banned. There was mostly only cocaine on the market. And so there was no more taxonomic revision that happened until Richard Schultes published this paper in 1957, where during the course of his extensive investigations in the field of human uses of plants, he described the preparation of cocoa, which actually involves taking leaves and pulverizing them, mixing them with some other leaves, and ingesting them that way, which is the standard method of coca preparation throughout the Amazon. So renewed interest from this real legendary botanist, and he assigned his student Tim Plowman to figure out what was going on with coca, what was the real diversity of coca being grown in South America, because it was widespread and poorly defined. So Tim Plowman started at Harvard, started studying cocas in the 1970s, going to the field at that time. And then he moved to the Field Museum, and he was a curator here from 78 until 89, when unfortunately at that time he died. He published 46 articles on Erythroxylum, including the modern taxonomy of coca. So here's what he has described, and this is this standing taxonomy. There are 2 species of coca, each with 2 varieties. So we have 4 total taxa of coca. All of them are allopatric, or mostly allopatric. The first, Erythroxylum coca, variety coca, Bolivian or Huánuco coca. Traditionally, this is the coca responsible for most traditional production by indigenous peoples on the eastern slopes of the Andes in Peru and Bolivia and up into Ecuador. And then there's Erythroxylum coca variety Ipadu, which is Amazonian coca. This one's remarkable because it's actually propagated vegetatively, which could have some consequences for the genetics. And the other 3 are propagated by seeds. Third, we have Colombian coca, which is in the other species, Novogranatense. And this one is remarkable because it's the only one that's self-compatible, meaning that pollen from a plant can be pollinated onto itself and it will produce viable seeds. The other 3 varieties do not do that. Lastly, we have Trujillo coca, which is Novogranatense variety Truxillense. This is grown in the very dry valleys of northwestern Peru. And these are the leaves with extensive archaeological evidence, probably because they're in, or certainly a factor in that is because they're in dry places. Most of those archaeological sites are in drier areas along the Andes. And these are the leaves still used in Coca-Cola. So taking a step back, coca plants, coca crops, these are shrubs. They have simple green leaves. They are used as ornamentals in people's gardens because really they look quite nice. They have small white flowers, which are hermaphroditic and pollinated by various sort of generalist insects, bees and wasps and flies. And I've seen all sorts of insects on these flowers. They produce small nectaries. And then the fruits themselves are single-seeded drupes that are 0.5 centimeter to 1.5 centimeters in size. And these are readily eaten and dispersed by birds. So yeah, there you have it. These are the 4 varieties. But But in order to gain a more in-depth understanding of what these things are, it's important to understand their evolutionary relationships. So what are the relationships between the two species? And then what are their closest wild relatives? Similar to humans and apes, it's like we need to put in context what coca is by identifying its wild relatives. However, this is complicated by the fact that coca, or very similar species to coca, are not found in the wild. So this is what Plowman came up with. He thought that the progenitor of the coca crops were actually it was a wild and now extinct form of Bolivian coca that grew on the eastern slopes of the Andes in the same geographic distribution as Bolivian coca today. After being domesticated there, in the early Holocene, Plowman thought that Bolivian coca was taken up the coast to the region where Trujillo coca is grown, and there it evolved into Trujillo coca, became adapted to drier areas. And then Trujillo coca was taken further north where it evolved into Colombian coca. So Colombian would be the most derived variety in this series. However, in addition to that series, Bolivian coca was taken into the Amazon where Amazonian coca evolved. And this is believed to have happened more recently, perhaps in the last 1,000 years, because of really homogeneous linguistics as well as methods of preparation and consumption of coca leaves throughout the Amazon basin. Emmanuel Johnson at the USDA was the next one, really the only other one to look at this question of the origins and evolution of coca. In '99, he showed that each variety actually has distinct flavonoids as well as leaf morphology. And so this sort of went counter to Plowman's hypothesis, because if there was a linear series of domestication, we might expect the chemistry in the leaves to be a subset, or at least similar across all of these things. Derived varieties would have a subset of chemical diversity as what was found in the progenitor varieties. And in addition, in 2005, they looked at AFLPs, where they found a clear differentiation between the species, coca and Novogranatense, and also between Bolivian and Amazonian coca. So in this plot on the right, you have Novogranatense on the left, and an intermixture of Trujillo and Colombian coca among those samples. But then on the right, in the triangles, we have the two varieties of Erythroxylum coca, and those are genetically distinct according to AFLPs. Also remarkable is the fact that Amazonian coca had less genetic diversity than the rest, which were fairly equal. So taking all of this into consideration, Johnson countered Plowman's hypothesis, though very respectfully, and said it doesn't really look like there's this nesting of Colombian and Trujillo within Bolivian coca. And instead, there could have been a single domestication of a common ancestor and subsequent allopatric diversification of each of these species and varieties. And so then with the start of my PhD, I was the first one to continue genetic analyses of coca. And I've done this in collaboration with Adolfo Jara Munoz, who is now a professor at the Universidad Nacional de Colombia. And our process here was to first identify the closest wild relatives via phylogenomic inference to the cocas. And we took a pretty broad sampling here. And then once we've identified those closest relatives, then we will more intensively sample the cocas and those relatives throughout their populations across their geographic ranges. And then we will, conduct various tests and analyses in order to look at these previous hypotheses. So the closest wild relatives of coca, this was not an easy thing to manage. There are a few previous hypotheses of a handful of species that are grown in the Andes, but yeah, nothing very convincing. So I also want to point out here, the sheer number of species of Erythroxylum in the neotropics. There are over 200. So yeah, I believe in Amazonia, this is, this genus ranks, this is some of Nigel Pittman and colleagues' work. This genus ranks as about number 20 in holding the highest species richness in Amazonia. And they grow in palm swamps, dry forests, as well as moist forests, and even into the Caatinga of northeastern Brazil, so real deserts. And then there are 75 species in the paleotropics, which also display a similar sort of ecological diversity. So for our genetic data, we assembled a draft genome starting in 2014. We had a mate pair library. This genome assembly is actually just recently published with Stacy Pirro of Iridian Genomes. But then our process with that draft assembly that we've had for years now and had years ago was to combine that with a transcriptome and identify 541 nuclear genes that were, we believed to be low copy. So these things were, conserved throughout all of these wild species, and thus they were good genes to analyze in order to understand evolutionary history. And so to figure out these wild relatives, we had 289 samples in the coca family, Erythroxylaceae. And this is what the picture of biogeography and biome evolution looks like. So we have the phylogenetic tree at the bottom. We have a global map at the top, and the character states at the tip of the tree start off with the world region for which each one of these tips, which is a species where that occurs. And we can see that the oldest lineages in Erythroxylum belong to moist forests in Central Africa. After this, we have a decent number of species occurring in Madagascar. And then into the Indo-Pacific, there's a handful of species in Australia, as well as many other countries to the north. But then we have a monophyletic clade of neotropical Erythroxylum. All right, and that is where most of the diversity lies. So those bars are in gray, all of those that occur in South America. They are brown for those that occur in Central America, Mesoamerica, and they are red for those that occur in the Caribbean. Right? And then above that, you can see the biome diversity, which I'm just going to kind of skip over here, but we have sort of a random distribution. Most of the species in both hemispheres are occurring in rainforests, but then there are numerous biome switches into savannas and dry forests, right? In the red box here, I have identified the coca clade of interest. So within this clade, we can identify the immediate wild relatives of coca. And there are two of those. In this phylogeny, on the right, you can see an outgroup, Erythroxylum fetidum, followed by Novogranitense, the two coca crop varieties in that. And then we have Erythroxylum cataractarum, which is a fully wild species, Erythroxylum gracilipis, and then the two clades of coca. So very confident that these are the closest relatives. And cataractarum, there are a few mentions in ethnobotanical literature of human uses of each of these, but more for cataractarum, where the leaves, and both of them do produce cocaine, though in very minute quantities compared to the crops. And yeah, people have been chewing cataractarum in the region of the Llanos and the upper, Rio Negro tributaries of Colombia and Venezuela. And then you can see in this map, highlighted in green, the distribution of Erythroxylum gracilipis, and it is really widespread. But an interesting distribution because it's kind of going around the heart of Amazonia. Okay. So now that we've identified those wild relatives, we've moved forward with target capture sequencing again using the same 541 genes, this time 144 samples. We assembled these gene targets with Hyde Piper called SNPs also. And then we are going to start off by inferring a phylogeny again. But yeah, really remarkable, and something that I want to point out here is that this was, to my knowledge, the first study that has relied mostly on museum samples for a large, global, systematic phylogenetic effort. So for that global phylogeny, 70% of the 260 Erythroxylum samples, there were more in some other genera. That's why I quoted 289 before. And then for the coca clade, which is shown in this map here, over 88% of the samples used in this analysis were from the herbarium, and the vast majority of those from the field museum. So this really demonstrates the utility of a well-curated museum collection, one where we have high confidence in species identification, high confidence in species numbers, and a variety of samples, which we can try and get DNA from, because you always are going to have to sample more than one per species in order to get good DNA. At least that's certainly my experience. So you can see in this map, the dots that are within the polygons correspond to each taxon in the analysis. And the point that I would like for you to take away from this is that even after adding in more GBIF data or herbarium records, the samples that went into this analysis from the herbarium pretty much allowed us to cover the entire geographic distribution of each one of these species, and thus have a much higher confidence in our inferences later on. Right? The DNA itself was mostly fragmented to less than 400 base pairs. I would also add that I usually sampled about 7 to 10 collections before getting one that had good DNA in it. But for those that have DNA in its degraded state, there's really very little correlation with the success of our gene assembly. So in these two plots, on the bottom left, you can see the year collected on the x-axis versus the fragment size of DNA molecules on the y-axis. The cluster around 1980 is all of Tim Plowman's work. And then these red X's are samples that had low input DNA quantity, although less than 200 nanograms, it's not really extremely low, though less than the protocols called for. And then as we use this DNA to assemble the genes, the figure on the plot on the bottom right shows us that the actual fragment size of your starting DNA did not have really any effect on the success of gene assembly. So we were able to piece back together these genes and use them in our later analyses just as well with some of the old stuff, or the real herbarium collections with heavily degraded DNA. Those work just as well as the new things that I'd collected in 4 or 5 years ago. So before we get to the results, let's touch back on these hypotheses. So Plowman's single origin hypothesis, where we have an original domestication of Bolivian coca, would manifest in a phylogenetic inference by having all of the cocas within a single group. So wild species would be exterior to all of the coca samples. And then we would see that Bolivian samples should be present in both of the clades, the Erythroxylum coca clade of Bolivian and Amazonian coca, as well as the Erythroxylum novogranitense clade. So we would have Bolivian, Trujillo, and Colombian. And furthermore, we would see a nesting of Amazonian, a nesting of Trujillo, and a further nesting of Colombian cocas within each one of their presumed progenitors, right? If we counter that with Johnson's sister species hypothesis, we would expect to see, again, just a single clade, a single monophyletic group of all of the cocas. But Erythroxylum coca, Bolivian and Amazonian would be in one clade, and then sister to that would be Trujillo and Colombian. But this is what we actually see. Coca forms 3 clades within the diversity of Erythroxylum gracilipus. So the colored clades here are collapsed clades. And in the top, in light blue, we have Trujillo coca. And next to that, we have Colombian coca. So that is a single clade representing the Novogranitense species. Traversing down the tree, we have Erythroxylum cataractarum being sister to other Grassolepis samples. And then at the bottom of the tree, we see the Amazonian clade is separate from the Bolivian clade. So therefore, we have 3 separate clades. By itself, this is, of course, very different from what we would expect if the scenario was what Plowman or Johnson had proposed. However, there's really poor support around the bottom of the tree. And so it is possible that if there's been introgression from Grassolepis, gene flow out in the wild, one way or another, that this separation of Amazonian and Bolivian could be due, it could be artifactual. And in fact, these things do form one clade. But the next step is to utilize the SNP data and assign individuals in this tree to populations. We can see that clustering identifies an optimum of 5 populations or 9 populations. What's notable from the 5 populations cluster, you can see at the bottom, is that Amazonian coca is actually green. So it's actually assigned with very high probability to Grassolepis, as opposed to Bolivian coca or its own clade, and it is assigned to its own population in the K = 9, the 9 population assignment. So in both of these also, we have Grassolepis broken up into multiple populations, and there is not much geographic structure in these population assignments. And what that indicates is that we really have some cryptic diversity in this widespread wild species. And that is, the subject of ongoing research, which could result in some new species being described. Also important, there are Trujillo coca samples that are actually inferred to be Colombian. And these are from a disjunct population in northern Ecuador. They are not from the general geographic area in northern Peru. Talk a little bit more about the mysteries surrounding Ecuador in a little bit. And lastly, if we look down at the Bolivian coca clade, we can see that some of the Grassolepis samples are actually red, and that means that they are inferred to be part of the population with Bolivian coca. And that indicates that either there has been introgression into these, or alternatively, this is a signature of ancestry. And these 2 samples from the Department of Madre de Dios in southern Peru are indicative of the region of domestication for Bolivian coca. But we can test admixture, a number of ways. In this case, I'm using tree mix. And so we're looking for admixture between Grassolepis and some of the cocas, because we want to see if there's evidence for problems in our phylogeny arising from, or problems in our phylogeny that would split Amazonian and Bolivian coca, right? We kind of expected them to be together. But in fact, the only evidence we have for admixture is into cataractarum. So in this plot on the left, in the top right, we have inferred admixture from Colombian into cataractarum. Or if we extend this and say that there was multiple admixture events, we see from Amazonian and Colombian into cataractarum. And then lastly, possibly from cataractarum into one of the Grassolepis populations. And so, the into cataractarum from the coca samples kind of makes sense because they're geographically proximal. And if there have been larger population sizes of the coca crops themselves, then natural introgression could be expected. However, nothing here really changes our intuition that Amazonian and Bolivian cocas are actually separate clades. So these are the outstanding questions moving forward. Are those 2 clades actually independently domesticated from Grassolepis 1? And then secondly, what is the progenitor relationship of these Colombian cocas and Trujillo cocas? Remember that Colombian is the only self-compatible variety, and this is, pretty confidently, pretty confidently going to be a derived trait because it is difficult to evolve in reverse. And so another question we should pose is which one of these has higher genetic diversity? So testing the number of origins, is there really are there separate clades for Bolivian and Amazonian is the real question here. So we used approximate Bayesian computation to simulate SNP data under 4 topological scenarios, which you can see along the bottom here. We have Plowman's domestication hypothesis, Johnson's hypothesis, and then we have a 2 origins hypothesis where Bolivian and Amazonian are within a single clade, or a 3 origins hypothesis where they're separate. And so what ABC does here is calculate the probability of topological scenarios by comparing observed versus simulated summary statistics. So summary statistics derived from SNP data simulated under coalescent models within each one of these topological scenarios. And in a nutshell, we use 2 different SNP data sets because each one of these data sets only consisted of 1 SNP per gene. So we were only analyzing in really about 420 high confidence SNPs in each set. And each one of those gave us a different answer. There was slightly higher support for 2 origins in one of those, from one of those comparisons with SNP data sets, and for 3 origins in the other. And due to those few numbers of genes and only wanting to sample 1 SNP per gene for each of those, this is where we left it with ABC modeling. Because we already have a very different picture of what's going on here with the cocas, and yeah, we need more extensive sampling to really get at this. But nonetheless, let's look at some other data. So a principal component analysis really supports 3 coca clades, because across these 2 principal component axes as well as subsequent ones, we never see really an intermediacy between Bolivian and Amazonian and then grassolipase 1, and we also never see really a nested relationship between samples of these varieties. And secondly, we also can't really tell from this whether Colombian or Trujillo is intermediate or nested and thus a progenitor. But as far as genetic diversities, we see that Amazonian is actually the highest genetic diversity, followed by Colombian, and then Bolivian, and then Trujillo. So as far as Amazonian being derived according to prior hypotheses, or Colombian being derived according to prior hypotheses, it's not that simple. And really, perhaps we shouldn't expect it to be. Yeah, and yet, if you remember Johnson's hypothesis, he found using AFLP data that the genetic diversity of Amazonian was actually the lowest of all of them. And so the sampling that they had there is fairly broad. There were maybe 25 samples, and yet their geographic origins aren't explained. And unfortunately, the plants that are still at the USDA, though much fewer in number, really don't have any geographic provenance information. So I think we can believe here that the sampling used in this study was more comprehensive, and thus gives us a better picture of overall genetic diversities across these crops. And then lastly, looking at genetic distances. So 3 coca clades are supported again, because each Amazonian and Bolivian are genetically closer to grassolipase 1 than they are to each other. And again, we can't really tell whether Colombian or Trujillo is more proximal to grassolipase 1 because they have similar genetic distances, right? So here's where we're at. This is a totally novel hypothesis. wherein we believe that there were multiple domestications of coca from Erythroxylum grassolepis. We have most support for 3 independent origins of coca crops. We have Amazonian, we have Bolivian, and then we have the Novogradatense clade, consisting of Colombian and Trujillo cocas. Our data also indicate that Bolivian coca was domesticated in southern Peru. And due to its higher genetic diversity and proximity to grassolepis 1 populations, the Amazonian coca is probably the most recently domesticated of all of these varieties. And that probably happened in Northwest Peru where they're, Northwest Amazonia, Ecuador, Colombia, Peru, where their distributions overlap. And yet there's no clear evidence as to what's going on within the Novogradatense clade. And this is because there has been really a long history. These are very old crops, and they have no doubt experienced a number of demographic shifts and intense selection. And so a simple, model of a bottleneck from the progenitor through a bottleneck into a derived crop is way too simplistic of a model in order to understand, certainly summary statistics of the genetics of these crops. So, and in addition, we have sampling gaps in Ecuador and Venezuela, and really a paucity of samples in those places. So moving forward, we need to get to Ecuador, which I did in 2021. I collected 38 specimens within this coca clade, including Colombian hybrids. There's a picture here from right on the Colombian border where this tree on the right-hand side is an old Trujillo coca. And Plowman visited here in the early 1980s. And I was very excited to see that a few of these trees are still there. However, they're being inundated with new hybrid varieties coming from the other side of the valley. So to the left of this tree, you can make out that other side of the valley, and you can see some green pasture land. All of that is illicit coca production on the Colombia side of new hybrid varieties, and the birds are carrying them over to the Ecuador side. And so, these old trees representing this more ancient variety of Trujillo coca, possibly Trujillo coca, is still there, and we were able to collect it, and that was really exciting. But as for Venezuela, I'm not really sure what we're going to do. Also moving forward, we'll be analyzing whole genomes, so a lot more data, and we're going to model multiple bottlenecks and add ancient and antique samples to the mix. Okay. And so with that, I'm out of time. Hopefully in the future, I can discuss some reflectance spectroscopy work. So with that, I would like to, again, point to the importance of our museum collections, and I'd be happy to talk about, the methodologies that I've been using, and hopefully we can, continue to improve these through time. So thank you to these other people as well, and thank you all for attending.
Hello. Okay. Do you have time for maybe one question?
What's the problem with Venezuela?
Well, we have very few botanical records. I mean, it's a, as I understand it, it's not something that I've really researched. It is next on the list of things to figure out, but the economic situation in Venezuela, the partnerships with herbaria there are going to be, they're going to be difficult right now. And but frankly, I don't really know the best way to go about field work there. Yeah, we have a number of Erythroxylum from Steiermark's collections there. Yeah, so it's well represented here. However, it is not well represented. Our collection here is not well represented for coca crops. So, Ecuador and also Venezuela are two places where coca eradication during the colonial period appears to have been much more successful. That's possibly because the Spaniards were not cultivating coca, managing the cultivation of coca, the production and the distribution, especially into mines where they were paying indigenous workers with coca leaves. So in Ecuador and Venezuela, our historical knowledge and our botanical collections are almost non-existent.
From the
Plowman was there. A bunch of wild species.
a few questions from the folks online. So we've got one from Udo Mireles. Question is, did native groups move coca around over long distances? And has that affect inferences about geographic origin?
Yeah, good question, Dudu. The understanding of routes of dispersal and trade networks are a really interesting dimension of understanding the significance of coca crops as well as the capacity of these ancient societies. However, with the sampling that we have based on, and that fills the maps that I've shown, these, in the time of Plowman at 1970, it seems that these were really restricted to allopatric distributions. They were all separate from one another. And thus far, we don't see much evidence for admixture, and we don't find Colombian coca in Peru or Bolivia and vice versa. So it would appear that, any sort of dispersal, any sort of trade, long distance trade of these coca crops in ancient times was only within what we understand their modern distributions to be.
We'll do one more before we let you go have a well-deserved lunch. So question from Bruce Patterson. Nice talk, Dawson. Great to see the big picture. So in animals, there are characteristic morphological and behavioral changes that accompany domestication, neoteny, fecundity, age at maturation. For example, besides cocaine concentrations, how do the cultivated varieties differ from their wild relatives?
Yeah, that's an important thing to demonstrate in domestication scenarios. And the coca crops all have smaller leaves compared to Grassolepis. The leaves are a little bit softer, and also, an interesting character that's been described is the more virgate or wand-like branches. So coca is harvested by running your hand up the length of a small branch and just ripping off all the leaves. And so while we have greenhouse grown varieties of coca, it appears that these things produce longer branches with long straight branches, as opposed to a more densely branched ramified state in Grassolepis. So we certainly have chemical differences with those samples in Ecuador. I'm in the process of metabolite characterization with colleagues down there, and higher cocaine concentrations, and then these smaller, softer, more palatable leaves, and apparently this difference in branching patterns.
All right, just want to thank everyone. For coming out today again. And for those that are here, we're going to go out to lunch with Dawson. So thanks again, Dawson. Have a great day, everyone.